Good afternoon, everybody. My name's Andrew Thorley. I'm a lecturer at the National Heart and Lung Institute, and I'm based at the Royal Brompton Hospital in Chelsea. And I'm going to talk to you today about some of the research we're doing in our group to try and come up with better ways to treat lung cancer. Now, there are many common cancers out there. The top most common ones that people tend to think of are things like breast cancer, prostate cancer, lung cancer, some of the other ones. I mean, what do you think is the most common cancer that's diagnosed every year? Breast cancer. Anyone else? Prostate. Lung cancer is actually the most common cancer. It's more cases of lung cancer are diagnosed every year than any other cancer. So around 1.68 million people are diagnosed with lung cancer every single year in the world. So it's a very significant disease, and the 5-year survival rate is quite poor for lung cancer at the minute. So a lot of people with lung cancer tend to present at a very late stage with their disease, and by that time the cancer has spread throughout the body. And that means that they need surgery and chemotherapy as well. But chemotherapy isn't very good and effective in treating lung cancer. And there's actually been quite a big shortfall in the funding to look into better treatments for lung cancer. So although it's the biggest killer worldwide and is the most common cancer, if we were to rank the funding for all the cancer research funding that is given every year, lung cancer is almost at the bottom of the league table for its research. So we were really trying to find some new ways to try and help treat lung cancer and get better improvement of the therapies. So many of you may know people that have had cancer and had to go through the process of chemotherapy. And the way that we traditionally give chemotherapy is we give it as an injection of a liquid into a vein, and then it distributes around the body. So if I just take this here, and this is a bag filled with a solution, and if we say that this is our blood vessel with the chemotherapy in it, and this beaker here is the whole body, if I put this in here, what you'll see is that over time, because that solution can pass through the permeable barrier of that membrane that I've put the drug in, very quickly it starts to leak out of the blood vessels and has now entered the whole body. So you can see that chemotherapy that was in that bag has now almost completely gone, and it's all distributed throughout the body. And that's why we get a lot of the side effects that we associate with chemotherapy. Things like hair loss, damage to the nerves, damage to our hearing, gastrointestinal disturbances. All these things are caused because the drug is allowed to go all over the body. So one of the things we're very interested in our group is, can we target chemotherapy drugs only to the tumor? So if we can get the drug to only go to the tumor cells, can we prevent all those side effects that really affect the patient's quality of life while they're being treated for their cancer? And also by being able to target the drug to the tumor, hopefully we'll improve the treatment of that cancer and improve the patient's survival rate. And one of the things we're interested in is using some of the biology of a tumor and taking advantage of some of the biological characteristics of tumors and using those to design our medicines in a better way to try and target them to the tumor. And one of the things we're interested in is using nanotechnology. Now, nanotechnology is the science of using particles that are 1/1000th the width of a human hair, so very, very tiny particles. And by doing that, we can take advantage of one of the phenomena that we see in tumors when they develop. So if we imagine this is our human blood vessel, in a very healthy, normal vessel, we have an intact wall. So whilst the drug solutions can diffuse through this, like I just showed you there, larger molecules, such as the nano-sized molecules, can't get through that healthy blood vessel. But when they get into the tumor blood vessels, because the tumor grows very quickly and it's very aggressive, the blood vessels in that tumor are very poorly organized. So the blood vessels are very leaky, so things can move out of the blood vessels and into the tumor quite easily. So that's where the nanoparticles come into play. And so if we take these as our example of nanoparticles, if we were to take our drug and put it onto a nanoparticle, when we put it into the blood system, pour them in, you can see that whilst they're in the healthy vasculature, these nanoparticles can't get out of the blood vessel. They're trapped. So if we attach our drug to the surface of the nanoparticle, when we put these drugs and these nanoparticles into the blood, when they're traveling around the healthy parts of the lung, they'll stay attached to the particle and won't be able to get into those healthy parts of the lung to cause the side effects. However, when they start to then pass further along and enter into the tumour, you can see where you've got that leaky vasculature, they can then start to move out into where the tumour is. But those that go past the tumour, again, they'll carry on and they won't go into the healthy cells, but only when they come back round again, they'll start to come out with the tumour. So this is a way of being able to selectively deliver our chemotherapy to the tumour. And it's hoped that by doing this, we can prevent any of the chemotherapy going to other parts of the body and causing the side effects that we so commonly see in treatment of cancer. And so Alex is now going to tell you some of the research we're doing, particularly in our research, in our laboratories on lung cancer in particular, and how we're designing the surface of these nanoparticles and what we put inside the nanoparticles to then target and kill the tumour cells that we see on that side of the vasculature.
Okay, hello everybody, I'm Alex. So as Andrew alluded to, I'm just going to try and basically explain a little bit more detail about the research that we're undertaking in our laboratory specifically. So there are two main approaches that we're interested in. The first one is gold nanoparticles. Okay, so in the spirit of Halloween, I decided to use a pumpkin for this prop. So the first thing that might come to mind is why gold? So gold nanoparticles are good for several reasons. The main ones that we found is that our research in our laboratory is found that these nanoparticles are actually very biocompatible, so they don't harm the human body like one would expect. Secondly, as well, once the tumours have accumulated, once the particles have accumulated in the tumour, as Andrew demonstrated earlier with the tube, you can use imaging techniques such as CT scans, that is a routine procedure done in hospitals, to allow you to get better images of the tumour as well. So not only can you deliver chemotherapy with these kind of particles, but you can also image as well. So our strategy was trying to attach two chemotherapy drugs to these gold nanoparticles. Their names are cisplatin and pemetrexed. So these drugs are the first-line therapy for a type of lung cancer that is the most prevalent, which is called non-small cell lung cancer. So our strategy for this was we use a special linker molecule, which is a type of plastic, and we attach this linker molecule to the nanoparticle, and then we attach our chemotherapy drug to the linker, and you get something that looks similar to this. And we repeat this many times. So we start to see a sort of covering over the nanoparticle of our chemotherapy. One other aspect as well that we sort of focused on with our gold nanoparticles is using sort of a more targeted approach. So as Andrew demonstrated earlier, that was more of a passive approach that just sort of happens as a natural phenomenon when you inject these particles. So another sort of area of research we're interested in was using special kind of molecules that target special proteins on cancer cell receptors that they are more abundant on these cancer cells than they are on normal cells. So as these nanoparticles enter the tumour cell via this method, we then take the same technique, we use our linker molecule to decorate the nanoparticles, and then we add our special targeting molecules to the surface, like so. So as you can see, we can deliver both a mixture of chemotherapy with the addition of targeting molecules to try and basically increase the efficacy of these drugs in the tumour, and also to stop the side effects that Andrew alluded to earlier as well. So the other approach we decided to take is sort of using a plastic-based material to create a different kind of nanoparticle. So the drug that we were interested in with this sort of research is one called vinorelbin, which is more toxic to the body. So when this drug is given as an injection, which it is for the kind of cancers that it's used to treat, you see that it's toxic to the blood vessels, and it also has other side effects as well, such as the gastrointestinal disturbances. So the approach we decided to take was to use So we have our drugs here, and we take our drugs and we encapsulate our drugs in these nanoparticles. So as you can see there, we have our drug molecules inside of these nanoparticles. So again, these nanoparticles will pass along in the circulation and then selectively go only into the tumour. And we can also, with these kind of particles as well, we can also attach our special molecules that target just cancer cells and not healthy cells to these nanoparticles to again try and selectively enhance the delivery of our drug just to the tumour and to leave the healthy cells as they are. I believe that comes to the end of our talk. I'd like to answer any questions and thank you for your attention.